Hello students, welcome to EPG Pathshala. I am Dr. Divya Bajaj from Hindu College, University of Delhi. Today we are going to discuss about the module Principles and Applications of Cross-Reactivity, Precipitation, Agglutination and SPR from the paper Immunology. After reading this unit, you will be able to understand the nature of antigen-antibody interactions, define the basic characteristics of an immune reaction, identify and define factors important for detection of antigen-antibody reactions, define the methods and principles of techniques like agglutination, precipitation and SPR, describe the various applications of these techniques, and describe the principles used in clinical applications of these tests. Starting with the nature of antigen-antibody reactions, we have the lock and key concept. An antibody molecule comprises of the FAB portion, where the active site consists of hypervariable regions of the heavy and light chains. The antigenic determinant resides in a cleft formed by the active site of the immunoglobulin molecule, as indicated by X-ray crystallography studies. Thus, the antigen-antibody interactions can be simulated by a key, which is the antigen. It fits into a lock, which is the antibody. It is characterized by formation of non-covalent bonds. The binding of an antibody and antigen is highly specific and involves weak and reversible non-covalent interactions comprising mainly of van der Waals forces, electrostatic forces, hydrogen bonding and hydrophobic forces. The antigen combines to the antibody at the active site by non-covalent bonds. Multiple bond formations ensure that the antigen will bind tightly to the antibody. Antigen-antibody interaction is reversible in nature. The antigen-antibody complexes are strengthened by non-covalent bonds, thus making their nature reversible. Talking about affinity and avidity of the antigen-antibody interaction. Affinity is the intensity of the reaction between an antigenic determinant and active site on the antibody molecule, defining affinity of that antibody. Affinity is the equilibrium constant characteristic of antigen-antibody interaction. It is the sum of attractive and repulsive forces between the antigenic determinant and the active site of a specific antibody. Most antibodies have extremely high affinity and specificity for their antigens. The avidity amounts to total strength by which an antigen binds to multiple antigenic determinants and multivalent antibodies. Avidity is affected by the valence of the antibody as well as that of the antigen and is therefore more than the sum of individual affinities. Hence, affinity is the strength of binding between a single antigenic determinant and its corresponding individual antibody combining site, whereas avidity refers to the overall strength of binding between multivalent antigens and antibodies. Specificity and cross-reactivity. Specificity is defined as the ability of a particular antibody active site to recognize and interact with only a single antigenic determinant, or the ability of a population of antibody molecules to react with only a single antigen. Antigen-antibody reactions possess extremely high degree of specificity. Antibodies are capable of discriminating between the primary structure of an antigen, isomeric forms of an antigen, secondary and tertiary structure of an antigen, hence being extremely specific. Cross-reactivity, on the other hand, is the capability of a particular antibody active site to react with more than a single antigenic determinant or that of a population of antibody molecules to react with multiple antigens. Cross reactions are multispecific interactions arising due to sharing of an epitope by a cross-reacting antigen and the immunizing antigen, or because the epitope is structurally similar to one on the immunizing antigen. Factors affecting measurement of antigen-antibody reactions. Antigen-antibody complexes are formed under specific conditions of temperature and pH. A number of factors influence the detection of these complexes, like affinity. Higher affinity of the antibody for the antigen ensures a stable reaction between the two, thus facilitating the detection of the complex formed. Next, it depends upon avidity. Interaction between multivalent antigens and multivalent antibodies are very stable, aiding in their detection. Next is antigen to antibody ratio. The formation of antigen-antibody complexes is related to the concentration of the antigen and antibody. Therefore, its detection is directly dependent on the ratio in which they are present at a particular instance. Lastly, it depends upon the physical form of the antigen. The physical form of the antigen plays an important role in detection of its interaction with an antibody. Particulate antigens result in agglutination on reaction with an antibody. However, precipitation of the antigen would occur in case it is soluble in nature when large insoluble antigen-antibody complexes are formed. This slide describes the process of agglutination. The word agglutination comes from the Latin agglutinare, meaning to glue, referring to clumping of substances. Agglutination is defined as the visible clumping of a particular antigen when mixed with antibodies specific for it, in the presence of electrolytes at an appropriate temperature and pH. Antibodies are capable of binding multiple antigen molecules, linking them to create a large lattice-like complex which is visible to the naked eye. Antibodies that generate such reactions are called agglutinins. All antibodies can theoretically agglutinate particulate antigens, but IgM, due to its high valence, is particularly a good agglutinin. Large antigens with multiple epitopes easily adhere to particles such as animal cells or bacteria when combined with specific antibodies, resulting in cross-linking. The process of agglutination involves two basic steps, the first being sensitization and the second lattice formation. Sensitization is the recognition and attachment of specific antibody to the corresponding antigen. Temperature, pH, and time of incubation influence this reaction. A lattice is formed by cross-linking between the sensitized particles. This figure demonstrates the process of agglutination, where a pentavalent immunoglobulin IgM is shown to interact and bind with multiple antigenic epitopes shown in color orange on the surface of bacterial cells, causing clumping or agglutination reaction. This slide demonstrates agglutination reaction in ABO blood typing. The serum of individuals would contain different types of antigens on surface of RBCs and corresponding antibodies to these antigens. The red blood cells from individuals of different blood groups are shown in this table. We can see that a person with blood group A has red blood cells demonstrating no antigen on the surface. A person with blood group A, for example, has antigens of type A on the surface of the RBC and so on. So, if serum from individuals of a particular type of antibody is introduced into a person with a particular type of blood group, reaction between antigen molecules on the surface of RBCs and the antibodies will occur, resulting in agglutination. The table here describes several combinations which are clearly seen. Instances where agglutination occurs and instances where agglutination does not occur. This slide demonstrates hemagglutination. Hemagglutination reactions involve agglutination reactions involving erythrocytes. These reactions are used in blood typing, diagnosis of certain diseases, and identification of certain viruses. Blood typing tests with the ABO antigens involve mixing of the RBCs on a slide with the antisera to the antigens A or B. If the corresponding antigen is present on the cells, agglutination results in the form of visible clumping on the slide. Identification of the antigens present on donor and recipient RBCs is the fundamental principle for matching blood types for transfusions. The figure shows red blood cells interacting with viruses to produce a clumping reaction which is clearly visible for all the sets. Now coming to what Coombs test means. When erythrocytes interact with antibodies, agglutination may not always result. This could be due to deviation from antigen-antibody ratio from its optimal concentration or due to zeta potential on the erythrocytes preventing cross-linking of cells. In order to detect the non-agglutinating antibodies on erythrocytes, a second antibody directed against the antibody attached to their respective epitopes on erythrocyte is added. This anti-immunoglobulin can now cross-link the erythrocytes and result in agglutination. This test is known as the Coombs test or anti-immunoglobulin test. The Coombs test is based on two important facts. 1. Antibodies of one species are immunogenic when injected into the other species, leading to production of anti-immunoglobulins. Second, many anti-immunoglobulins bind with an antigenic determinant present on the Fc portion of the antibody and leave the Fab portions free to react with antigens. This slide shows the process of precipitation. Precipitation reactions in fluids yield a precipitin curve. When a constant amount of antibody is taken in a series of tubes and increasing amounts of antigen is added to these, variable amounts of precipitates form, resulting in a quantitative reaction. Initially, this method was used to determine the amount of antigen or antibody present in a particular sample. After precipitation, the tubes are centrifuged to pellet the precipitate, and the amount of precipitate is measured on removing the supernatant. The amount of precipitate, when plotted against increasing antigen concentration, yields a precipitin curve. Excess of either antibody or antigen interferes with maximal precipitation. Hence, formation of an insoluble antigen-antibody complex occurs within a narrow optimal concentration range, known as the zone of equivalence. This zone represents the condition under which antigen-antibody complexes formed are sufficiently large to be precipitated. At equivalence, a large multimolecular complex is formed which increases in size and precipitates out of solution. On the other hand, outside this zone, antigen or antibody excess occurs, resulting in the formation of small soluble complexes. This slide demonstrates the formation of the precipitation curve. It shows the interaction of antigens with specific antibodies that generate the precipitate. Different zones are highlighted in different colors according to the concentration of differently distributed components. We can see that the antibody in precipitate is shown on the x-axis and the antigen added in increasing concentration is shown on the y-axis. The zone of equivalence is shown in blue and it represents the maximal optimal concentration of antigen in antibody interaction. The precipitin curve is shown in red. Coming to the technique radial immunodiffusion. Radial immunodiffusion is a simple quantitative technique where a serially diluted antigen present in wells diffuses into the gel matrix containing uniformly distributed antibody. When the antigen diffuses into the gel, it reacts with the antibody as soon as it encounters the latter. This is followed by attainment of the equivalence point visualized in the form of a ring of precipitation around the well. The diameter of the precipitation ring is directly proportional to log concentration of antigen, since the amount of antibody is constant. Thus, the amount of an antigen in an unknown sample can be quantitated from a standard curve obtained from different concentrations of a standard antigen. If more than one antigen-antibody reactions has occurred, corresponding number of multiple rings would form, indicating that the sample contains a mixture of antigens or antibodies. This test is commonly used for determination of immunoglobulin levels in clinical samples. This slide shows radial immunodiffusion. The precipitin ring formed as a result of antigen-antibody interaction is visible in the agar matrix in the form of a radial sphere. The antigen molecules are present inside the well in the center of the agar matrix and the antibody is uniformly distributed within the gel matrix itself. The antigen molecules migrate radially and as they migrate, they interact with the antibody molecules to form antigen-antibody complexes and the precipitate ring is formed at the optimal concentration. This reaction can be easily visualized with the naked eye. This slide describes immunoelectrophoresis. Immunoelectrophoresis involves placing a complex mixture of antigens in a well and electrophoresing it on an agar gel so that the antigens are separated according to their charge. Following electrophoresis, a trough is cut in the gel and antibodies are added to this trough. The antibodies diffuse into the gel, resulting in formation of precipitin lines in the equivalence zone as a consequence of the antigen-antibody reaction. This test provides qualitative analysis of complex mixtures of antigens along with a crude measure of quantity represented by the thickness of the precipitin line. The components in a patient's serum are commonly analyzed using this test, where serum is placed in the well while antibody to the whole serum is placed in the trough. Comparison with normal serum can determine whether there are deficiencies or excess of one or more serum components on visualizing the thickness of the precipitin line. This slide describes the mechanism of countercurrent electrophoresis, also called double immunodiffusion or Ochterlony. The Ochterlony test, named after the Swedish physician who invented it, is one of the most commonly used serological preparation reactions and is based on double immunodiffusion. Also known as countercurrent electrophoresis, it involves placing the antigen and antibody in wells punched in a gel matrix and allowing the two to radially diffuse into each other, hence producing a concentration gradient. Different geometrical patterns have been observed in the precipitin lines formed between the antigen and antiserum wells. The patterns of these lines are indicative of the amount of similarity or dissimilarity between the antigens. Rates of diffusions of these molecules increase in proportion to their concentration in the wells but decrease in proportion to their sizes. Only antigen and antibodies having opposite charges can be used for this reaction. This test is primarily quantitative, although semi-quantitative analysis can be performed by analyzing the thickness of the line. Its major advantage is its speed. This slide shows the double immunodiffusion. The antigen and antibodies diffuse towards each other in gel matrix until they interact to form the precipitate in the zone of equivalence. This zone of equivalence can be easily visualized by naked eye, and the migration of antibody and antigen molecules can be seen when they move out into the gel matrix. This slide shows the different types of precipitin lines formed as a result of the Ochterlony test. Different patterns are produced when different types of antigen-antibody interact in the gel matrix. We can see a curve-like precipitin line when the antigen-antibody molecules are identical. The patterns form a crisscross pattern when they are non-identical, and when they are partially identical, we have a combination of both these patterns produced in the third set. Coming to the technique surface plasmon resonance or SPR. SPR is a non-invasive optical technique that involves label-free means of real-time tracking of binding of an injected analyte into an immobilized biomolecule. Analyte binding leads to a subsequent increase in mass, and therefore proportional increase in refractive index is observed as a shift in the resonance angle. The SPR procedure involves a flow injection comprising the analyte of interest dissolved in a buffer, traversing the sensing surface to network with the immobilized biomolecule. SPR transducers operate by a prism coupling incident light onto an optical substrate coated with semi-transparent noble metals like gold. Total internal reflection occurs at this surface such that all of the light is reflected while none is refracted above a certain incidence angle. First, TIR should occur at the interface until the incident angle drops to the critical angle at which a fraction of the light is refracted across the interface. At the point of reflection at the interface, a standing wave would penetrate the exit medium to the order of 1/4 of the incident light wavelength. This slide demonstrates the mechanism of the technique surface plasmon resonance or SPR. The basic principle of the technique is highlighted in this diagram. Coupling of the incident light with the surface plasmons affects the intensity of the reflected light and the patterns are analyzed. Figure A over here shows the light source which is incident on the sensing surface which is coated in this case with the metal gold. The optical detector captures the reflected light and the patterns of intensity produced are then analyzed. The binding of an analyte molecule leads to increase in mass and proportional increase refractive index is observed as a shift in the resonance angle. So students, let us summarize what we have learned in this module. The antigen-antibody interactions depend upon 4 types of non-covalent interactions like hydrogen bonds, ionic bonds, hydrophobic interactions and van der Waals interactions. Cross reactions are multispecific interactions arising due to either sharing of an epitope by a cross-reacting antigen and an immunizing antigen, or because the epitope is structurally similar to one on the immunizing antigen. The interaction between a particulate antigen and an agglutinating antibody, also called agglutinin, produces visible clumping or agglutination that forms the basis of simple rapid sensitive amino acids. The interaction of a soluble antigen and a precipitating antibody in liquid or gel medium forms an antigen-antibody precipitate. Electrophoresis can be combined with precipitation in gels in a technique called immunoelectrophoresis. Precipitation reactions are based on the interaction of antibodies and antigens, two soluble reactants that combine resulting into one insoluble product, the precipitate. These reactions involve formations of lattices or cross links. SPR is a non-invasive optical phenomenon that allows label-free means of observing interactions occurring during binding of an injected analyte into an immobilized biomolecule in real time. Thank you.